Okay, so this is the exam 4 neurolab review. What I'm going to do first, though, is sort of tie in some class concepts because then it'll make just our lab identification make a little more sense. And we're going to need the lecture review anyways. So here is a diagram. I've only put red arrows for the things that we've been focusing on in class, so this is what you need to pay attention to. Let's start up in our frontal lobe. We've got our big 3 eye fields. We have our frontal eye field. That's going to be doing our voluntary saccades, our horizontal and our vertical conjugate gaze. We're going to get our information coming from the cortex from the frontal eye field. That's going to be Brodmann's area 8. And it's going to be on this middle frontal gyrus, sort of on the lateral side of the frontal lobe. Then we have a little more anterior and inferior. Still technically Brodmann's area 8, it's just going to be sort of forward and down. From the frontal eye field is our dorsolateral prefrontal cortex. So dorsolateral, a little down and sideways from the frontal eye field. And that's going to be what's focusing on those, like honing in on the saccades that you're wanting to look at, and then the ability to ignore unwanted saccades. Like if something draws your eye, you have the ability to sort of choose not to have that reflexive gaze towards it. So that's just what that dorsolateral PFC is going to be in charge of. And then the supplementary eye field, which is actually sort of more medial on the frontal lobe, it's Brodmann's area 6, and it's going to be doing memory-guided saccades. So if you knew someone walked into the room, then you would have your eyes able to go to them, not having been keeping track of them while they've been moving out of your sight. So your eyes can just click to where you know they should be. That's going to be your supplementary eye field. So the big player for actual, the conjugate gaze pathways we've learned, it's going to be the frontal eye field. Then coming back to that parietal lobe, we have the parietal eye field. That's going to be in Brodmann's area 7. And that's going to be what's in charge of smooth pursuit. Technically, smooth pursuit and voluntary saccades are both sort of between frontal eye fields and parietal eye fields. But for ease of memorization and for Dr. Brewer, she wants us to know that saccades are frontal eye field and smooth pursuit is going to be in our parietal eye field. And that's just going to be the ability to, exactly what it says, just smoothly move your eyes in tandem. And then lastly on this, I have the superior colliculus, and that is going to come into play with reflexive tracking of something moving in your field. So if you have your something coming into your vision from your left side, so that's traveling from left to right, what's going to pick that up in your vision first is going to be your right-sided hemiretinas. And so then that's going to go back to your right superior colliculus, and then it's going to travel with your crossed tectospinal tract to go over to the left, that left PPRF that is going to then direct your horizontal saccades towards your left to sort of see that thing coming into your vision. Okay, so I'm really quickly going to review conjugate horizontal gaze, and then we'll go over vertical. So for horizontal, you're going to be getting contralateral input from your frontal eye field. So that's going to cross over and activate your PPRF, which is a paramedian pontine reticular formation. That is going to be your brainstem instigator of horizontal conjugate gaze. So when your frontal eye field hits that, it's going to activate your abducens nucleus. And so that nucleus is both going to stimulate the ipsilateral lateral rectus to look the left eyeball to the left. And then it's going to have those other set of neurons that cross, go to the contralateral MLF to reach this oculomotor nucleus, which then can do our medial rectus to have this right eyeball look to the left. Then you have your conjugate gaze. The whole time this is happening, we have our maintenance keeper because we want to maintain this process so we can continue looking to the left. So when you ever hear maintenance, that is a buzzword to mean either your nucleus prepositus hypoglossy, if we're talking about horizontal gaze, or INC, if we're talking about vertical, but we'll get to that. So all this NPH is here to do is to just keep sending reflexive signals. They're going to be communicating back and forth to say, hey, keep going, keep looking, keep looking. Really quick to talk about lesions, so let's just go through these really quick. If you take out this nucleus, well, you've taken out the ability for this left eye to look left. You've taken out this ability for the right eye to look left. So you have horizontal gaze paralysis towards the side of the lesion. You cannot look this way now. Abducens nerve, if you cut right here, well, your contralateral eye can still look to the left, but your ipsilateral eye no longer can. So that would sort of look, if someone had this lesion, you told them to look to the left, this eye would stay forwards, this eye would look to the left, they would look a little cross-eyed. Now say you take out the MLF. So if you take out this MLF here, same concept, one eye is working, one eye's not, but it's switched. So now this one's looking to the left fine, this one's staying. So now if this guy was told to look left, he would keep this eye forwards, this eye would look left, and his eyes would be kind of farther apart than they're supposed to be. Now, 1.5 syndrome, that's if you take out this abducens nucleus and then also the ipsilateral MLF that's not drawn in here because I'm showing you just left gaze. So if we took out the nucleus and this MLF, so first of all, we know that taking out this nucleus means this eye cannot look left anymore. Well, if we also took out the MLF coming to this same eye, this eye can also not look to the right if it wanted to look to the right. So that's the one part of the 1.5. The ipsilateral eye is completely horizontal gaze paralyzed. However, the half part comes from this eye because since we took out this abducens nucleus, obviously we don't have the medial rectus having this eye look to the left, but its own abducens nucleus is working fine, so it could still look to the right when it wanted to, right? Because if you activated the lateral rectus from here, it would look to the right, it'd be fine. So this one has a half paralysis because it can't look left, it could still look right. This eye is donezo for horizontal anyways. And then just a quick note for the frontal eye fields, because it's just contralateral information, if you took out this frontal eye field, you would have temporary paralysis to the opposite side. But because there is some overlap just in the cortex, it would eventually sort of correct itself a little bit. But yeah, so let's move on to vertical real quick. Okay, so I separated these to downward and upward. Just a real quick overview because the lesions are the more important part here. You're going to get bilateral frontal eye field input. So like both of these are going to be putting in, I just showed one side on this diagram. So bilaterally goes to both RIMLFs. So this is the brainstem instigator of upward and downward, so vertical conjugate gaze. So then this RIMLF is going to send signals to both the oculomotor nucleus and the trochlear nucleus. So that way, the oculomotor nucleus can go and activate the inferior rectus of this eye to look down. And then also, so that this trochlear nucleus can go and cross over contralaterally and activate the other eye's superior oblique. So the superior oblique being activated means the eye is going to look down and out. This eye is going to look down and in. But the reason that you have the conjugate vertical gaze downward is because at the same time this is happening on this side, you have that bilateral input. So the same thing's happening here, but in reverse. So when you activate this one, it's going to activate these nuclei. 3 is going to come over here and do the inferior rectus here. So when you combine superior oblique and inferior rectus, you get a perfect down gaze. And then like I said, your INC is going to be your maintenance keeper here. So that's going to be super important. It's going to sort of just communicate ipsilaterally between all of these nuclei to make sure everything continues to go. And then let's go on to the upward gaze. Okay, so conjugate upward gaze, a little more complicated. That's why we're really only focusing on the lesions, but just to go over it. So imagine you're getting that bilateral input to your RIMLFs. Your RIMLF this time, instead of just saying hey to the ipsilateral oculomotor nucleus, it's actually going to send from one side and activate both of your oculomotor nuclei. And then those are going to activate both muscles that it takes to look upward. So you have your superior rectus to make you look up and in, and then your inferior oblique to make you look up and out. Combine those, you're looking straight up. So your RIMLF is doing that to this nucleus and also this nucleus. Now your INC is still doing that maintenance, but the way it does its maintenance is different for vertical gaze. Instead of just talking to everybody on this side, it actually crosses over and talks to this side, and same thing over here, just to make things harder. And then all of this crossing over is happening in the posterior commissure. So the big problem with vertical upward gaze is that if you take out this posterior commissure, a lot of this stuff is relying on bilateral communication. So that's going to be an issue. So if you could get bilateral upgaze palsy, meaning both eyes cannot look up, if you have, say you have a bilateral RIMLF lesion. Because if you just took out one, you would still have this other side available to talk to both sides' oculomotor nuclei. So you would really have to see a bilateral lesion to be able to take out enough to get your actual upgaze palsy. Another way would be to block the artery of Percheron, which not everyone has. This is if you're missing, instead of two arteries supplying this like region in the posterior commissure, some people have one artery of Percheron that supplies this area. So if they had that and it got blocked, you would then lose the ability of the PCOM. You wouldn't have blood supply here, and then none of this would be working. And then also is blocking the posterior commissure. You remove the blood supply from it or you block it, same deal. And then this Perinaud syndrome is important. So Perinaud syndrome is this blocking of the posterior commissure. It's usually via hydrocephalus or a pineal tumor, because the pineal gland is going to be sitting right over here. So basically anything impacting the PCOM crossing can cause this Perinaud syndrome. So what you see when you block this is obviously you have the bilateral upgaze palsy or supranuclear gaze palsy, because you're not creating this palsy by taking out a nucleus. It's actually above the nucleus, so this PCOM. You get bilateral eyelid retraction, convergence retraction nystagmus, pupillary light reflex impairment. So just keep track of all of these deficits you're going to have if you do block something, instead of necessarily looking step by step through this diagram. Okay. So here is a quick little thalamus overview slide. I have a feeling that borders are going to be a big thing, so I just wanted to really quick review the borders of the thalamus. So let's look anterior border. This is going to be right where your foramen of Monroe are, so like right here. That's going to be your anterior border. Your posterior border is technically, they say, around the area of the posterior commissure, though it does jut out past it at some points. So that's more of a theoretical posterior border. Medially, and since we're looking at it from a mid-sagittal section, like this surface of the thalamus medially is bordering on the third ventricle. So you would have your third ventricle space right here. And then laterally, you have your genu and posterior limb of the internal capsule. And that makes more sense when we look at cross-sectional images. But when we see in those lateral ventricles, when they start opening up and you have your thalamus pressing up against the lateral ventricle and the third ventricle, the other side of that is going to be your internal capsule. So then superiorly is the lateral ventricles, so that's obviously this space up here makes sense. And then inferiorly makes even more sense, thalamus, inferior hypothalamus. And then a quick little note is that in the thalamus, 75% of those neurons are projection, excitatory, glutamate, and the other 25% are interneurons, which are going to be those inhibitory GABA. Okay, and so here is this thalamus diagram. This is more just for you to take a look at in your free time. Free time, I say lightly. And this diagram has really helped me when I'm looking at cross-sections and I can't understand why I'm seeing what nuclei I'm seeing. I just imagine drawing a line straight through, and then I can see what I should be able to label on a cross-section depending on how forward or back I am. So just save this slide and you can look over the functions of everything. But for the sake of time, I will just move on. Okay, and I also believe that this will be very important because a great way to ask questions about, like, do you know the thalamus is, okay, say someone comes in with a certain problem, what area of the thalamus would be damaged? So we will go over this really quick. So if someone comes in with anterior grade amnesia or attention problems, you know that, okay, this must be a part of the limbic system because we're talking about memory here and making new memory specifically. And so that should make you think, okay, Pepez circuit. Well, big part of that is the anterior nucleus getting information from the hippocampus via the fornix and the mammillary bodies. So if you take out this, you've taken out that circuit. Therefore, you cannot make new memories. So that would be the anterior nucleus. Another, specifically the dorsomedial nucleus, also can cause anterograde learning issues because it is receiving, it's also part of like this limbic sort of association cortex. So you're receiving all these association cortices and you're presenting out towards those associational prefrontal cortex. So you can have difficulty anterograde learning. So learning specifically has a big thing to do with the dorsomedial nucleus. And then looking back here, this one makes a little more sense. Your pulvinar is in charge of your visual perception. So not necessarily like actual sight, but perceiving what you see. So you're not going to see blindness with difficulty or a lesion to hear, but you're going to see lack of attention to visual stimuli as a deficit. Now we know these, obviously, MGN's your auditory relay, so auditory deficit's going to be there. Visual reflex deficits specifically, and tracking because of that superior colliculus communicating here, you would have issues with your lateral geniculate nucleus. Your VPL and VPM are going to be your somatosensory issues. So VPL is that body contralateral somatosensory, so pain and temperature. VPM, also contralateral, except for taste. But you're going to see loss of pain and temp from the face because the VPM is for the face. And now VAVL, this is in charge of motor stuff. It's getting a lot of input from the basal ganglia and the cerebellum. So because it is getting a lot of cerebellar input, you're going to see your cerebellar deficits. So if you see, contralateral ataxia of the limbs or intention tremor, bradykinesia, anything that we saw as a problem with the cerebellum in our last lecture, because our cerebellum is directly talking to these two, now we could see those same deficits manifest by a lesion here. Okay, so now we're going to look at the thalamus from a little different angle. Here's the thalamus. We're looking down on it from sort of a Heimer dissection. So our thalamus here, we can see this is about where the foramen of Monroe is. See opening into the third ventricles. And then our PCOM is hidden right here, but that's going to be about our posterior border. So our caudate nucleus is going to have our head here and extending back, and our head and extending back, because our thalamus is going to be located medially and inferiorly to the caudate nucleus. We also know that on cross-section, our caudate nucleus is the lateral border of the lateral ventricle. And so that makes sense because here you can see that, and you can see it bordering on this, this airspace, which is the lateral ventricle. An important groove that these make because they border each other, the caudate nucleus and the thalamus, is the thalamostriate groove. So that is the line between the caudate and the thalamus. The thalamostriate groove contains the thalamostriate vein, which runs here, but what runs over top of the vein are axon fibers making up the stria terminalis. And the stria terminalis is going to be linking the amygdala. So we're going to show that a few slides away. It's going to be linking the amygdala to other limbic structures. So that's going to be an important player. And then, so that's going to be sort of our lateral thalamus stria. We have a medial thalamus stria thing going on over here. We can't see it because it's going to be like truly running across the middle of the thalamus. So like if we were looking at this little piece right here, this is sort of sitting on top of the medial side of the thalamus. It's going to be deep to this. It's going to be our stria medullaris thalami, and it's way easier to see on cross-section. The stria medullaris thalami is going to be linking your hypothalamus to your brainstem. And then also we will see our habenula, because our stria medullaris thalami, as they run back, those stria are going to end up as a habenula. So they're gonna run and then bulge out and become two little habenula that you'll be able to see on this next slide. Okay, so here is a better picture. So here we have our thalamus again, our caudate nucleus. Here is our thalamus striate groove with our thalamus striate vein running through that, stria terminalis over top. Then we have here, finally, we can see our stria medullaris thalami. And that's going to be running on the medial surface of the thalamus, which we will come to see is our dorsomedial nucleus. And this is what I meant when it sort of builds back into this habenula, and habenula here. And this is your pineal gland. And then you can also see really well the massa intermedia, which is going to be this section connecting the thalami. Oh, and before I forget, also, this is a really good place where you can see your anterior tubercle. So this little notch or bump coming off of the front of the thalamus is very, very noticeable. And this is where the anterior nucleus is going to underlie. But it does make a really nice little bump off the top of your thalamus. Okay, so now we're going to get into a cross-section here. We have a really good view of the mammillary bodies, so we know we're somewhat in the middle-ish of the brain. I have circled here the massa intermedia that I just pointed out in the last slide. Here's that connecting the two thalamus on either side. Now let's take another look at our thalamus striate groove. So I said that the caudate nucleus is the lateral wall, the lateral ventricle. So here you can see this is your caudate, because here's your lateral ventricle. And we know this is our thalamus. And I had said that the border between the thalamus and the caudate is that thalamus striate groove. So this little area right here is your thalamus striate groove. So this may be the lumen of the thalamus striate vein, but it's a little big and blown out, so we'll see some better ones on following slides. But let's say if this could be the lumen of your thalamus striate vein, then overlying it would be your stria terminalis. Okay, and then we also can see right here these black dots right here. So it's an axon stain, so these are axon. And what is a fiber bundle running on the medial surface of the thalamus? So it is the stria medullaris thalami. And that is what I had shown going back and creating those habenula back towards the pineal gland. And then as for our thalamic nuclei, we can identify right here. So far, we can see our dorsomedial nucleus. That's going to be pretty much present medially the whole run of the thalamus. And then we can also see this area is probably going to be our VL area, because we're not anterior enough that we're going to be seeing our VA. This is going to be more into our VL area. And then down here, we don't have any much like distinguishment between VPL and VPM, but that's where your VPL, VPM are going to be. And something else is, here's your mammillary bodies. Here are some really obvious axon fibers leaving out of here. That is your mammillothalamic tract. So this is a really good picture of the mammillothalamic tract. And that's going to be these mammillary bodies communicating with the anterior nucleus. And this actually makes a really good point, because the mammillothalamic tract is connecting to the anterior nucleus. So why do you not see these axons traveling up to this nucleus right here? That's because this is your dorsolateral nucleus. So these are your dorsolateral. Typically up here, you would see your anterior nuclei, but you're only going to see the anterior nuclei up here in front of, anterior to these mammillary bodies. Once in cross-section you hit the mammillary body, it's going to start being the dorsolateral nucleus. You've transitioned too far back, and now it's turned into that instead. Okay, so here is some just bookkeeping on the different regions. So thalamus versus hypothalamus and epithalamus. So you have your thalamus here. Your epithalamus is going to be constituted of your habenula and your pineal gland. So here's your pineal gland. Your habenula would be on either side of the pineal gland. You can't really see them here. So this sort of area is going to be your epithalamus. Here's your thalamus. And then you know your hypothalamus is underneath the thalamus, but you can actually see a really good hypothalamic sulcus right here. So that's going to be the delineation between thalamus and hypothalamus. You can't always see it super well, but this is a really good picture of it. So everything below that, this is going to be your hypothalamus. And then to talk about recesses, basically any recess is named for what it's right by, and a recess is going to be a divot inward. So let's say optic recess. We find our optic chiasm. It's going to be our duck bill of our little duck guy. So bill, eyeballs, the anterior commissure. Here's its whole little head. It's looking to the left, puffing out its pons chest. So you find your optic chiasm. This little divot is your optic recess. Okay, and then you have your pineal recess. So find your pineal gland. Here's that. This is your pineal recess. Infundibular recess and stalk. So optic chiasm is going to be right in front of the infundibulum. So it's kind of cut off here, but you can see the remnant of the stalk right here. It's meaning that this is your infundibular recess, just where it divots in a little bit. Now we also need to find our mammillary bodies. Those are going to be right here. So here's the mammillary bodies. The lamina terminalis is the front of that duck's face. That's going to be the anterior-most border of your neural tube. It's going to be derived from the telencephalon. Dr. Smith probably will never ask a question on it, but it seems like he will, so you should learn that. And then, like I said, for the duck eye is the anterior commissure. And then one thing that I find pretty hard to see is your tuber cinereum. This is supposed to be another bump coming out, kind of like your anterior tubercle. However, this one is supposed to be posterior to the optic chiasm and continuous with the infundibular stalk. So this right here should bulge out. So you should be able to see an appreciable bump. But because it's kind of hard to see, just memorize where it's supposed to be. So it's supposed to be continuous with this infundibular stalk and just posterior to the optic chiasm. Okay, so here is another cross-section. Let's check it out. So let's see where we're at. I see big red nuclei. I see pes pedunculi, substantia nigra, so we're pretty far back in the brain. And I can also see a little bit of the pineal gland right here. On cross-section, the pineal gland, you can see it a lot better than this if you were to cut it a little bit further back. So because of that, because you can see sort of air space in between here and it's kind of not very darkly stained, you can tell that you kind of have just scraped into it and haven't quite gotten back there yet. Another way you can see that you're pretty posterior but not quite to the very posterior is that you can see your lateral geniculate nuclei pretty well, but your medial geniculate nuclei is barely peeking out, because imagine riding a roller coaster back like towards this cross section. And as the cross section progresses, we are sitting right at the point where the cross sections start being cut, like if you were to go down the roller coaster. So if you were to cut a little bit at an angle, like you were sitting on a roller coaster looking down, that's when you would see the median geniculate nucleus. It's like we haven't gotten over the crest of this pes pedunculi yet, because once we get over this, we'll have a view of the median geniculate nucleus. So now that we know where we're at in the brain, let's again point out a few things. So here's our lateral ventricle. Here is our caudate nucleus. So caudate, thalamus, thalamostriate groove then. Here's a thalamostriate lumen, and our stria terminalis is laying over top. Now our habenula, like I said, here's your pineal gland. So on either side, you can see the habenula really nicely, okay? And then you also can see from the habenula, you have some axonal fibers running down. So that is actually communication from the habenula to the interpeduncular nuclei. And so here's your peduncles. So interpeduncular nuclei down here. The easier name for this tract is the fasciculus retroflexus. However, the longer name is probably better to learn because it literally has in the name where it's going. Habenulo interpeduncular fasciculus. So that it's really best appreciated on cross-section. And then we can also see for our thalamic nuclei, we can point out our dorsomedial nucleus pretty well. We can also see our lateral posterior nucleus. And this is a big reason why it's important to see like that we're not completely as far back as we could be based on these two little signs here I talked about. Because if we were all the way far back, then this nucleus would be the pulvinar. But because we haven't quite gotten back far enough in the brain yet, we are just in front of the pulvinar, which is the lateral posterior nucleus. But let's get a zoom in and we'll look at the rest of the thalamic nuclei. Okay, so this is the same cross-section, we're just closer in. So like I said, this is your lateral posterior nucleus because we haven't quite reached the pulvinar yet. We still have our dorsomedial nucleus going strong. And at this point, we're far back enough that this little area here where we have our VPL, VPM, we can distinguish between the two. So here, medially, obviously, our VPM, and then this other area of sort of darker colorage is our VPL. So something to note about the VPM is that it does sort of, it's said to like crescent shape over this nuclei. It doesn't quite look like that to me, but it does sit up against it. So just remember, if you see a little circular nucleus, sitting next to a bigger, more appreciable nucleus, that's going to be your VPM. This nucleus you can see is within the thalamus. It's not really a border of anything. That's going to be your centromedian nucleus. Same over here, centromedian, VPM, VPL. And then also, so our centromedian nucleus is going to be one of our intralaminar nuclei because it's within the medullary lamina. The other intralaminary nucleus that's basically associated with the centromedian is the parafascicular. And that is just this lighter portion of the circle. This is your parafascicular nucleus. This is really the only cross-section that you can distinguish them very well. So I imagine if we were asked to distinguish, it would be on a cross-section like this where you can see that delineation. And then our internal medullary lamina is what is making our intralaminary nuclei. Our external medullary lamina is this black going on the lateral border of our thalamus. But you can see all these little dots, these little lighter colored dots. That is going to be our reticular nucleus. And so that's the nucleus that's just doing interneuron projections to the thalamic nuclei, inhibiting them, and basically gatekeeping the information that's going around in the thalamus. And here, again, we can see our LGN, and this black axon fiber going and up against the LGN is going to be our optic tract, because our optic tract is going to be feeding back to that LGN. Okay, and here's our little internal capsule summary slide. So I have the longitudinal view where you're looking over top of the brain of the whole internal capsule, and then I have 3 cross sections progressively getting from anterior to posterior in the brain. So anterior, you're going to have communication from the anterior nucleus upward. So that's the only thalamic nuclei that uses the anterior limb. All of our other thalamic nuclei are going to use the posterior limb. Okay, so you have anterior, genu, posterior, and then when you get behind this putamen, the lentiform nucleus, it's called your retrolenticular internal capsule. And if you were to look beneath this nucleus, you would see your sublenticular internal capsule. So you can see that pretty well here. So here's your lentiform nucleus, and here's your sublenticular internal capsule, this black fiber right here. And so looking at these cross sections, you can tell you're anterior here because your putamen and your caudate are merging to form the nucleus accumbens. When it looks like owl eyes, it looks anterior to me. So here's your anterior limb then. This also is where you're going to find frontopontine fibers. And then let's go to this next section. We're still pretty anterior because they still look kind of owl eye-ish. Now we've reached the anterior commissure. But the reason I included this, since it's pretty close to the anterior, is now that you can see the foramen of Monroe. When you see foramen of Monroe in the cross section, that means that the part of the internal capsule you're looking at is the genu. That's just exactly where it lines up, because here's where your foramen of Monroe is. You cross-section that, you are going right through the genu. And then let's go back here. We see thalamus. Then that means once we see thalamus, that's going to be our posterior limb. And that's where you're going to have your corticospinal fibers coming from. So all of your sensation and motor fibers coming down. And then one more note is that in the genu, the one thing about the genu is that your corticobulbar fibers are going to be the closest right there. And then what you're going to find in your retrolenticular internal capsule is you're going to be finding your optic radiations. You're also, which, yeah, you're going to be finding your optic radiations there. But then on the sublenticular internal capsule, so back here or underneath, you're going to be finding auditory radiations and also Meyer's loop optic fibers. So all the other visual fibers are going to be found retro, but your Meyer's loop is going to be found sublenticular. Okay, and so here are some internal capsule deficits. So let's say you take out the anterior limb. Like I said, the anterior limb has communication from the anterior nucleus of the thalamus. It's the only thalamic nuclei that communicates through the anterior limb. So if you damage this, you've cut off the anterior nucleus communication, which is the Pepez circuit, like I said. So there you've got memory deficits. You cut off here, that's where your auditory radiations and your Meyer's loop. So you could get auditory deficits, or you could get contralateral superior quadrantanopia, pie in the sky, Meyer's loop. You see your posterior limb. You have any somatosensory, any spastic paralysis from your motor of the contralateral side, upper and lower limb. You're going to be seeing posterior limb deficits or a lesion there because you have upper and lower limbs all communicating through this same little pathway. And then if we had a damage to the the retrolenticular, you're going to see contralateral hemianopia because all the rest of your visual fibers are coming back in this area. Okay, so now we're going to start looking more at the thalamic nuclei just to review. So first things first, let's see where we are in the brain. So I'm seeing just the very start of the thalamus starting to come out. So we're getting posterior, but we're right where the thalamus is starting to emerge. That said, the first thing that you're going to see on the thalamus is the anterior nucleus. You can also tell because we have a really appreciable bump right here. So that would be your anterior tubercle on a gross specimen. So that's your anterior nucleus. We also can see a hypothalamic sulcus, which is really nice. It divots in like this. So there's your thalamus and then bordering your hypothalamus. And then your dorsomedial nucleus. Let's look over to our cross-section a little further back. Here's our dorsomedial nucleus. Here's our anterior nucleus. And then here we are up pretty, we are in the thalamus, we're pretty anterior, so I would say this is VA/VL. And then down below we have our VPL, VPM would be starting. Maybe, I don't think we're quite there yet though. I think really we're gonna see the VA/VL. If we look back at the Thalamus diagram that I posted a few slides ago, and you kind of slice through where you're supposed to be on that sideways diagram, it'll make more sense why we haven't quite reached the VPL, VPM yet. And something else to look at here is this is really hugging around the anterior nucleus in that Y shape. So that's going to be your internal medullary lamina. It's easy to confuse the internal medullary lamina with the mammillothalamic tract because the mammillothalamic tract is going to be communicating to this anterior nucleus from the mammillary body. So I don't think we're going to be asked to distinguish between them, especially because the mammillothalamic tract travels through the internal medullary lamina. But Dr. Smith did say when you can see it very clearly hugging both sides of the nucleus, it's probably going to be the lamina. Whereas the mammillothalamic tract, you're going to see more of a straight shot towards the nucleus. Okay, so here we are at the mammillary bodies. So what does that mean? If we're at the mammillary bodies, what is this top nucleus? It's not going to be the anterior nucleus because we've reached the mammillary bodies. So now it's the dorsolateral nucleus. We do have a very good view of our mammillothalamic tract coming off of those mammillary bodies. We still have our dorsomedial nucleus right in that medial area. We have our little stria medullaris thalami. And we also have our caudate, and then our thalamostriate vein with our stria terminalis over top. And now let's look at what other thalamic nuclei we have. So now we've gone back, we're around VL now, because now that we've gotten mammillary bodies, we've probably surpassed VA. So now we're pretty much in VL territory. And now we're just now seeing our VPL, VPM start to appear, okay? Once we get further back, we'll start to see a delineation, like I said, between VPL and VPM. But for now, I would say just as a little grouping. And let's see, anything else? We have our basal ganglia. We already covered that, though. We don't need to rehash old wounds. Okay, so now we're pretty far back. This is the side we've looked at before. We have the just barely there pineal gland. We have our habenula. We have our lateral posterior nucleus instead of our pulvinar. Dorsomedial still going strong. Now we have that VPL, VPM, like I said. So here's the VPM, VPL, centromedian, parafascicular. Those are our intralaminar nuclei. And then a little fun extra note is you can see the lumen here of your internal cerebral vein. Okay, so now this is what I was talking about with how it seemed like in the last slide, your pineal gland wasn't all the way there yet. It's like you just barely scratched the surface. So here's where you can see it really well. You can also see the big posterior commissure. So now you know you're very far back in the brain. You can also see that we've gone over the hill, seen over the horizon of this pes pedunculi. Now we can see the MGN really well. MGN, LGN. And now, if you'll see, here's your lateral ventricle. It sees backwards and comes out into your temporal lobe as your temporal horn of your lateral ventricle. Here's that lumen of that. The roof we know is the tail of the caudate. Here's the head, or not the head at this point. We're far back, so this is just the body seeing around, becoming the roof of the temporal horn. Now we also have the floor of the temporal horn is our hippocampus, and we will be talking about the hippocampus parts here shortly. Okay, so here's another good view of our internal medullary lamina. We see this Y shape, so we have this coming up, delineating medial from lateral. We have some separation here, not sure exactly where it's going, but just you see that line, that's going to be our internal medullary lamina. And we also, again, have a really good view of our mammillothalamic tract. If you ever see axon or dark lines coming out of the mammillary bodies, it's your mammillothalamic tract. You also have a good view of your internal medullary lamina from above. Here's that good V line coming out just like that. Okay, here is a quick little blurb on blood supply. Right side is the long version. So you see here that you have each delineation of the posterior cerebral arteries. You have your tuberothalamic doing most of your anterior stuff. Then you have your paramedian, obviously going around doing some medial stuff. Thalamogeniculate is doing a lot of this back here. I remember thalamogeniculate because your geniculate bodies are in the back, so it's doing some posterior stuff. And your posterior choroidal is named with the word posterior, so that's also posterior. The easier way is you have a bunch of branches from your PCA are doing sort of a lot of your thalamus. And then the anterior tip is taken by this tuberothalamic, which is actually coming off of your posterior communicating rather than your PCA. So that's the big difference here. So if you were to lose tuberothalamic input, you're going to cut off blood supply to this anterior part. And just like we know the anterior nucleus is memory, if you took that off, you'd have memory issues. If you take off PCA supply, you're losing basically everything else. So you'd have maybe okay memory, but you'd have some sensory motor issues. Anything else that you can find in the function of any of the other thalamic nuclei would be taken out with that. So now let's do Lab 11 little bookkeeping. Okay, so this is our Lab 11 review. So this is our last lab of Block 2. A lot of these things we've seen before, just now we're looking at them in a new context, or at least with some context. So first we have our parahippocampal gyrus, and that is going to be the most medial gyrus on the bottom side of our temporal lobe. So you can see that right here, specifically this folding. And it's not just this guy right here, because this is actually our uncus, and it looks like it's almost a separate little gyrus because of how deep this is. But this little streak is actually just a really well-defined intrarhinal sulcus. So basically your parahippocampal gyrus is one sort of whole being, and then the uncus is that bump that we know of that sticks off, and it is delineated. It's still part of the parahippocampal gyrus, but this little folding that separates it is that intrarhinal sulcus. And then another gyrus we've seen before is the cingulate gyrus, and so here's our corpus callosum. Our cingulate gyrus is that gyrus overlying it. So this is that cingulate gyrus. Deep to that, those axon fiber tract, that's going to be the cingulum. And then also, let's look back down at that uncus here. Deep to the uncus is our amygdala. And then also just another limbic structure that we needed to have some housekeeping on is the mammillary body. So this is a fiber dissection, so you can see things look kind of weird around here because all of the flesh has been sort of chiseled out to get these fiber pathways pretty visible. But the mammillary body is actually going to be right here. And so the mammillary body has a connection with the hippocampus, to the parahippocampal gyrus, and the hippocampus via the fornix. And so you can actually see the fornix coming from mammillary body up, and then here's your fornix. It's going to swoop around and end up down into that parahippocampal gyrus. Okay, so this is sort of like a Heimer dissection to the max, and basically we've just isolated the hippocampus out of the brain. So it's kind of hard to orient yourself to this structure, but here's anterior, posterior. These are going to be sort of parts of your temporal lobes. And you can see here, this entire little formation here is our hippocampus. And this is the best picture that I've seen of the pes hippocampi, which is the most anterior part of the hippocampus, looking like a cat's paw, like Dr. Smith says. So here are the little cat's paws. Those are your pes hippocampi, and then this whole little structure here is the hippocampal formation. Okay, so now taking a look at some cross sections, let's go ahead and orient ourselves with some stuff we already know. Let's look for our lateral ventricles here. Lateral ventricles, these are going to C-shape, posteriorly and then arch around into that temporal lobe. So here is that little hole delineating the temporal horn of the lateral ventricles. And here's that other side. And so what we already know is that our caudate nucleus does the same thing. Our caudate seeds back and then comes, and actually the tail of the caudate becomes the roof of the temporal horn. But now what we're learning that's new is sort of that the hippocampus is becoming the floor of the temporal horn of the lateral ventricle. And the hippocampus is sort of, the parahippocampal gyrus is folding in on itself and sort of spiraling. And so the spiral has different parts that have different names. So the part that's the most inferior, so like just starting to get curved into this curve, is the subiculum. So sub, beneath, so subiculum is this portion that's like folding in the first. And then the subiculum transitions into the hippocampus proper, or the hippocampal gyrus, and this is sort of this arch up and around here, and then it ends in the middle as the dentate gyrus. So the dentate, as Dr. Brewer said, is the middle of this hippocampus jelly donut. And so we can look over here closer. This is going to be our, our screen left side of the brain. So we have our subiculum folding up into our hippocampus proper, and then our middle little dentate gyrus right here. And now some parts that are sort of part of the hippocampus but are also named as parts of the fornix, which we'll look at in a minute, are the alveus and fimbria. And so the alveus and fimbria are axon fibers, so you'll see them stained darkly here. And basically it's coming from the subiculum and parahippocampal hippocampal gyrus. And it's these black axon fibers here arching up over top. So this little thin line is the alveus. And then the alveus, you can see thickens, and this little bulb that it sort of thickens into is called the fimbria. So on the side too, you have your alveus and then your fimbria. Okay, so now we're looking at our inferior view up at the brain. So let's again point out our parahippocampal gyrus. Like I said, it is that medial most from the inferior view temporal lobe gyrus. So here's your temporal lobe. So let's look most medial. We have this gyrus right here. And you can see your uncus really well, this bump right there, and then the little line of your intrarhinal sulcus. Now there are also a couple more regions of this parahippocampal gyrus that now we can see. So I'm going to hop over to this image because we have got it zoomed in. So uncus, parahippocampal gyrus. So your entorhinal cortex is going to be basically lateral to the uncus. So here's your uncus. So then this area is going to be your entorhinal cortex. And this cortex is actually one of the main afferents into the hippocampal formation. So it's going to submit information to the subiculum, and then it's going to take it through that pathway, and it's also going to help consolidate memories. It's going to shoot back to the CA1 area to sort of consolidate memories, the end of that circular pathway. It'll sort of shoot back to the end and like basically tie up some strings. So entorhinal cortex. Then you have your piriform cortex, and we've heard this because we remember the piriform cortex is that primary olfactory cortex. And so the piriform cortex is the anterior most tip of the parahippocampal gyrus. So it's going to be right here. This area is your piriform cortex. So you'll have them here. And then your entorhinal cortexes. Uncus, amygdala is deep to that. And then I just have listed here the hippocampus afferents. So you have a lot coming from the Cingular Gyrus. You have a lot coming from the parahippocampal gyrus, specifically, like I said, the entorhinal cortex. And then you also do have some coming in from the amygdala. Okay, so this is a fornix slide. I've got a lot of different ways to look at it because it's kind of confusing. It's kind of C-shaped, just like a lot of the other things in the brain are. So the fornix, like I said earlier, it connects the mammillary bodies to the hippocampus. So we have mammillary body, fornix, hippocampus. So it's just a fiber pathway. So it's not too complicated, but it has three named portions of it. So we have the body is just the main stretching anterior to posterior area. Then they come down in the front and turn inferiorly, and these are the columns, and they're kind of close together here. They V off and separate into these columns towards the front, and then they do the same thing. They V off and come down towards the temporal lobes, and these are the crus of the, you can't see those obviously, but these are the crus of the fornix. And then again, I had pointed out the alveus. So the alveus, because we really pointed it out on the hippocampus, it's going to be like these fibers here, the alveus thinning, and then it thickens here and sits on top of the hippocampus as the fimbria. And then just a little FYI, the alveus is composed, those axon fibers are being made from, like I said, the subiculum and the hippocampus proper or hippocampus gyrus fibers. And then the fornix is actually suspended it's hanging up from the septum pellucidum. So here's your septum pellucidum. In this gross image, the septum pellucidum has been carved out, but you see that this is what was holding the fornix up from the corpus callosum via the septum pellucidum. And then also the hippocampal commissure is something we've had to point out a few times, but it's really hard to see because it's not very well developed in humans. But the hippocampal commissure, if you more just memorize the location of where it's supposed to be, that's really what you need. So the place where you're going to find the commissure of the hippocampi is going to be on the fornix, and it's going to be right basically at the border between the body and the crus, so at this bend. But the best way to remember that is that bend is basically on the anterior border of the splenium of the corpus callosum. So literally right here is going to be that hippocampal commissure. And yes, like I said, it lines the splenium. Okay, so now we have some more cross sections, our best friends. We've seen these a billion times, but now we're going to point out something new on them, the fornix. So Here we can see, are we really anterior or are we really posterior? Okay, so I'm seeing a really big pineal gland, posterior commissure, big red nuclei. This looks like midbrain right here, or brainstem, I mean. So we're pretty posterior. And we see where we have our lateral ventricles. So here's our corpus callosum. And we see these little things V'ing out really far apart. This is going to be our crus. So when we're naming parts of the fornix, it's really just taking into account how far forward or back you are in the brain, because you know up in the front, you're going to have your fornix separate into columns. One way to distinguish those is just, are you far forward or are you far back? But also in the back, they're going to V out way wider because they're trying to end up reaching that temporal horn. But in the front, the columns are just V'ing apart. So here's super anterior, or, well, you know, not super anterior, but anterior enough that we still have a really good view of our optic tracts, and then we're just at the beginning of our thalamus here. You can see our columns of our fornix coming down right here. And then we actually still see a bit of our septum pellucidum hanging that up, and then our columns are just V'ing out. And then our body, I have a slide coming up of our body that shows it just kind of looks, you know, a lot flatter, like you're not seeing that V, you're not seeing the separation quite as much. I'll show you in a couple slides. But let's see what else I need to point out here before we move on. Oh yeah, I'll just point out the alveus and fimbria again. So we have the black lining over the top of the hippocampal formation. That's the alveus, thickening into the fimbria. Same over here, alveus, fimbria. And then a little throwback, we've got our subiculum, hippocampus proper, dentate. And then if we look over here on our little anterior view here, we have our foramen of Monroe. So we're pretty far anterior. Here is our anterior nucleus. Here's our dorsomedial nucleus. And this is probably anterior enough, it may be that this is our VA. If not, VA, VL is good enough to distinguish those. Here's a good view of our cingulate gyrus and our cingulum, those black fibers within that. And then let's look back over here. We have our MGN and our LGN. Any of these black fibers sort of going up to that LGN is going to be our optic tract, because like we know, the optic tract is sending its fibers to that LGN. And then a throwback to the last lab is that we have our thalamus striae vein right here, that lumen. Over top of that is going to be our stria terminalis, and then this little groove is our thalamus striae groove. Okay, so here's some more fornix. Let's look where we are in the brain. I immediately see the big anterior commissure because we have this A, like we learned. The A is coming down, and what lines it is that anterior commissure. Well, now to add to that little way to remember, these parts of the A coming down are actually our fornical columns. So those are going to be the columns right there. Here's our septum pellucidum holding it up. And another ways just to solidify that we're anterior is we don't have the thalamus showing yet. This is our caudate head. And so this means that this is our anterior limb of our internal capsule. And below here looks like this is going to be our putamen and our globus pallidus externa, but not necessarily vital for this exam. Moving over here, this is what I was talking about, the fornix body. These are really close together. They're not really V'ing off anywhere. We're just kind of following this straight back through the brain. So this is when it's taking its long anterior to posterior path side by side. So here's the lateral ventricles, corpus callosum, cingulate gyrus is cut off a little bit, cingulum within that. We have a good view of our mammillary bodies, so we know we're sort of like, we're somewhere in the region of not super anterior or super posterior. The mammillary bodies aren't necessarily in the middle of the brain from anterior to posterior, but I always imagine, okay, I'm somewhere around the middle-ish. But also, check this out. So our mammillary bodies we have a pretty good view of. We also have a pretty good view of this structure right here. Now, this little line, you can see just barely connecting our mammillary body all the way back to this nucleus. So the trouble with identifying this little track right here is that when we see the mammillary body and we see something coming off of it, it's probably going to be our mammillothalamic tract. I'd say most of the time, because it's mammillary body to the thalamus, and the mammillothalamic tract is connecting that mammillary body to the anterior nucleus. But I did make a really good point, which is still true, that when you are cut straight through the mammillary bodies, like once you get to the mammillary bodies, this top nucleus you're going to see on the thalamus is actually the dorsolateral nucleus because we've gone past the anterior nucleus. But this may be an exception, not an exception per se, but because you can see, I think that this cross-section has been taken like when they just barely have scraped into this mammillary body. Like I think that there's a lot of mammillary body left behind this cross-section. So that said, we are right on that transitional zone between anterior and dorsolateral. And because we see this perfect tract, I'm going to have to assume that this is the mammillothalamic tract and this is that anterior nucleus. But then there's also this internal medullary lamina that is going to occupy essentially the same space because they sort of actually end up running together. Like the mammillothalamic tract is going to use that internal medullary lamina as like a highway to get to that nucleus. So I don't think that we'll be asked to differentiate between them because I think that's a little mean, but a way to tell the difference per Dr. Smith would be if it was going to be our internal medullary lamina, it would be a lot thicker and you would really see it grabbing the sides here. Like you would be able to delineate the Y shape a lot more. So I think that this is just going to be our mammillothalamic tract. Okay, so let's orient ourselves as we always do. I'm immediately saying anterior because here's our nucleus accumbens. That's my go-to. When I see owl eyes, this looks like an owl eye with its little eyelashes here. That means we're super anterior because we are so anterior that the putamen is reaching around and meeting via the nucleus accumbens that caudate nucleus. And so that is what I'm looking for. Now, granted, since we're so far anterior, we have a new little player we want to take a look at. So we have our septum pellucidum, and then we would have our fornix, but we're actually a little bit in front of the fornix. And although these look a lot like fornical columns, the best way to differentiate these columns from this being the septum pellucidum coming down into these septal nuclei, which is what this is, there are two ways. First of all, we have these lumen right here. These are our septal veins. So meaning this would be still part of the septum, but also our septal nuclei and that formation here is completely continuous with the nucleus accumbens. So like you can see here, it's not like it's sitting up against it. It is literally a part of it. Our fornix is not going to be it's not going to be molding into any structure like that until it gets down to the mammillary bodies. So when you see something continuous up towards the front and you're like, okay, is it fornix or is it septum? It's going to be septum. So like I said, here's where your septal nuclei are going to sit. They're kind of thickening out towards the bottom for these nuclei. There's your septal vein lumens. Here's your thalamus striate vein lumens. And then our fornix actually does project to the septal nuclei, and it's going to be sort of posterior to this formation. It's going to be projecting to these. And then also, since we are anterior, this is our anterior limb of our internal capsule. And then since this was our thalamus striate vein, here's our caudate. Over top of that vein, again, is the stria terminalis. And then this is going to be our thalamus striate groove location. And then also, we can see these black fibers going underneath the lentiform nucleus, aka putamen, and then our nucleus accumbens. This is going to be our sublenticular limb of our internal capsule because this is our lenticular nucleus per se. And so then these fibers underneath are sublenticular. So here we have our basal forebrain. This is an area more just a theoretical area than a place to easily point out. It's about the same location as the anterior perforated substance. So you remember that from like lab 1, it's on either side of the optic chiasm in these little pockets right here. So this sort of whole location is your basal forebrain, and that's going to be a big part of the cholinergic system because we've got our basal nuclei of Maynard that are going to send some acetylcholine. So if we look from the side, it's going to be about this area. So we're seeing the corpus callosum sort of beneath that. And then here's our mammillary bodies just sort of above. So just this general location and the basal nucleus of Maynard, I don't think we'll have to ID very specifically, but more just the general location. Then the ventral pallidum is like a delineation of the basal forebrain. And the ventral pallidum is specifically just the most dorsolateral region of the basal forebrain. So if we imagined the dorsolateral, so like the sticking out, there'd be this part of it. Here we would see like essentially this part of the basal forebrain would be our ventral pallidum. Okay, and then our amygdala. So our amygdala is, like I said earlier, deep to the uncus. It also is right on the tip of our hippocampus. So the best way to see it would be to point it out on a gross specimen. So let's look here. We have our anterior commissure, our fornical columns. So typically, when we look down here to our parahippocampal gyrus, we would be seeing our hippocampus. However, this is like the one time where that's not necessarily the case because we are so far anterior that we are actually cross-sectioning basically right here. So we are hitting through that amygdala. We haven't reached the hippocampus yet. You can tell because you see just like this is a lot of gray, and you don't see that little infolding where you could point out the subiculum and the dentate gyrus. There's no little spiral. This is literally this is the amygdala proper. Like this whole thing is just the amygdala here and here. And I think that might be one of the only times, I think what we're more likely to be tested on is sort of asking what's deep to the uncus, and you can also kind of see the uncus bump right here. That's another good way to point that out to yourself. But I think it's more important just to realize that it's deep to the uncus and be able to point that out on a whole specimen and just know that typically our hippocampus is going to be right here. But if we're anterior enough, look and see if we can point out the different convolutions of the hippocampus. If not, we're probably anterior enough where we can see the amygdala. Okay, so here's our lateral olfactory stria, and so this is pretty much was thrown in at the end of the lab, which was kind of funny. Like it seemed like it didn't really fit, but it's kind of a throwback as well because we've seen this a billion times. So we have our olfactory bulb and our olfactory tract, and we remember from our last exam that it actually separated into two little pieces on this tract. We had our lateral olfactory stria and our medial olfactory stria. So you can really see that here. So here's our tract, and we see this little V. So our medial olfactory stria is going to be crossing actually the anterior commissure to trade sensation with the other side olfactory stria in that anterior olfactory nucleus. But the lateral olfactory stria was actually where that smell was going to be transmitted to the primary olfactory cortex, which we remember is that piriform cortex. And where is the piriform cortex? It is that anterior-most tip of the parahippocampal gyrus. So here's that gyrus, here's our uncus, here's our piriform cortex, and look, our lateral olfactory stria is headed right for it. Literally going straight towards it. And then here we can also see the same. So here's our medial olfactory stria out of sight beneath the chiasm, and then here's our lateral coming straight towards this piriform cortex. And here's our uncus, here's our entorhinal cortex, here's our intrarhinal sulcus. And yeah, you guys are totally ready to crush the practical. And I hope everybody does super well, and I hope we're all excited for spring break.